this video, we're going to talk about action potentials and graded potentials. This is when we send electrical impulses down the axon so that we can release neurotransmitters and we can have signaling within our brain and from our central nervous system out to the periphery. How these impulses are transmitted is all about ions. So before we start talking about action potentials, I want to quickly review ion concentrations in a normal resting cell. In this cell, I have isolated a few ions that I want to focus on. First, we have sodium. There is more sodium outside of the cell compared to the inside of the cell. There is more potassium inside compared to the outside. In a previous video, we talked about how the sodium-potassium pump maintains this gradient by moving sodium out and by moving potassium in. And then we have chloride, which is higher concentration outside the cell compared to the inside of the cell. Now, the other thing that is significant here, other than the concentration of the ions, is the charge around the cell membrane. The cell membrane has a slightly negative charge on the inside and a slightly positive charge on the outside, primarily because there is more positively charged sodium outside of the cell compared to the positively charged ions inside the cell. Now, this relative difference is about 70 millivolts. So the inside of a resting cell that is not having an action potential is about 70 millivolts. And this is all cells in the whole body, not just neurons. Now we have what we call an electrochemical gradient. Electrical gradient because of the charge difference, and chemical gradients because of the differences in these ions. Now, let's suppose we open an ion channel. Let's use sodium as an example. If there is a sodium ion channel here, and we open that channel and we allow sodium to move across the membrane. Remember, the inside of the cell is negative and the outside of the cell is positive. Which way will the sodium move? Will it move in or out, and why? Sodium is going to want to move in because of the concentration gradient. There's more sodium outside than compared to inside. And also because of the electrical gradient. Sodium is a positively charged ion, and the inside of the cell has a negative charge. So a lot of sodium would want to move into the cell. If we opened a chloride channel, which way would the chloride want to move? So it would also move in because of the concentration gradient, but not as much as sodium because the charges, the negative charges, would repel a little bit. And then how about potassium? Let's suppose we open a potassium channel. Will potassium want to move into the cell or out? Because of the concentration gradient, it will want to move out. But again, not as much as sodium because the charges. There is less of an electrical gradient compared to the concentration gradient. So it is important to know which ions are in higher concentration outside the cell compared to the inside of the cell. And then it is also important to know that when we have this charge difference, a little bit positive outside, a little bit negative inside, about minus 70 millivolts inside the cell, that electrical difference means that the cell is polarized. Polarization is important because when we have an action potential, we are going to depolarize the membrane by moving ions into the cell. Then we have to repolarize the membrane and make that electrical difference occur again. So then that is repolarization. The next thing that is important to know is the difference between a voltage-gated channel and a chemically gated channel. Let's suppose we have a potassium channel here. And when it is closed, potassium ions cannot cross the membrane. And when it is open, potassium ions can cross the membrane. What makes this channel open? What will allow that ion to travel across the membrane? If it is a voltage-gated channel, then when there is a change in the charge, let's suppose we make this side of the membrane more positively charged, then if this is a voltage-gated channel, then the charge difference will make that ion channel open. If it is a chemically gated channel, then a chemical can make it open. Let's suppose a neurotransmitter binds. Neurotransmitters are signaling molecules that impact other neurons, and how they impact other neurons is by opening ion channels. So a chemically gated channel opens because of a chemical like a neurotransmitter, and a voltage-gated channel opens because of a charge change in the membrane. So now, where our ion concentrations are, the charge around the membrane, and our ion channels that allow ions to move. Okay, now that we have that information all sorted out, we are going to start looking at an action potential. When we look at a neuron, different things happen in different parts of the neuron. Here we have a neuron, and we call it a presynaptic neuron because there is a synapse in between it and the next neuron, which we call the postsynaptic neuron. If there was another neuron here, then this one would become the presynaptic and that would become the postsynaptic in this chain of transmission. Here we have a cell body and the dendrites. This is where information is received, like over here. If we have neurotransmitters that are going to be released onto the next cell, then these neurotransmitters can bind to ion channels on the dendrites. So the dendrites in the cell body receive the signaling information. So what kind of channels would we have here on the dendrites? Would they be voltage gated or chemically gated? They are going to be chemically gated ion channels. Now, what occurs in this dendrite cell body area is called graded potentials. Okay? The graded potentials are going to be the stimulus that will cause the action potential. Action potentials occur along the axon. The action potential direction is always in this direction. It's always from the dendrites in the cell body end towards the axon terminal end. And the axon terminals release the neurotransmitters. So we are going to focus on the action potentials first. In this diagram, we are looking at the charge change that is going to occur in that cell membrane as we have an action potential. So if this is our axon, there is normally more sodium on the outside of the cell and more potassium inside the cell when that membrane is resting. And the charge that occurs inside the cell, inside this axon, is about minus 70 millivolts. So this is our resting membrane potential. Now, in order for an action potential to occur in the axon, there has to be some stimulus, a triggering event. And this is basically the graded potential, which I'm going to explain in more detail in a minute. So this graded potential has triggered an action potential. Now, for an action potential to occur, this charge change difference has to be significant enough that it reaches a threshold level. If it's a very weak signal and the charge change only moves about 5 millivolts, then an action potential will not occur. Action potentials are considered all or nothing because they either happen or they don't. And action potentials are always excitatory. They have the same amplitude every single time. So now let's suppose we've had a triggering event, a stimulus, that brings the cell membrane to this threshold level. Let's look at our axon. Here's our axon membrane right beside the cell body. Cell body is here. The very first part of the axon is called the axon hillock. Now here in this area it is very rich in ion channels that are voltage gated. So whenever we change the charge of the cell membrane, it is a voltage change. So the stimulus that brought the resting membrane potential from -70 to about -55, that charge change will trigger these voltage-gated channels. So the voltage-gated channels are located all along the axon. Now remember, sodium is in higher concentration outside, potassium is higher concentration inside. The charge of the cell on the inside is negative. When this stimulus triggers these voltage-gated ion channels, it is going to make these sodium channels open. When the sodium channels open, sodium is going to move into the cell, because we just talked about how, because of the concentration gradient and the electrical gradient, when these channels open, sodium will want to move in. When sodium moves in, it further changes the charge, which is going to trigger more voltage-gated ion channels. This positive charge is going to trigger more sodium channels to open. Now this is going to continue to propagate all along the axon. We are changing the cell charge from negative on the inside to positive on the inside. So this is depolarizing the membrane. This early stage from the threshold level, when sodium ions start moving in, sodium is going to move in. This is depolarization. The membrane is going to become about plus 30 millivolts on the inside. So we've changed the polarity of the cell from -70 to plus 30. Now, once the membrane reaches plus 30, the charge change is going to trigger other ion channels that are voltage gated. The other ion channels that will be triggered are going to be potassium channels. Potassium channels are going to open because of this positive charge change. Now, which way is the potassium going to move? When a potassium channel opens, the potassium is going to want to move out. As the potassium moves out, it is bringing positive charges outside of the cell. When positive moves out, it is going to make the membrane negative again. This phase is called repolarization. So during the repolarization phase, potassium is moving out. Sodium moves in to depolarize it. Potassium channels are going to open, and potassium is going to move out, and that is the repolarization phase. Now, the potassium channels stay open a little bit longer than necessary to bring it back to exact resting membrane potential. So there is always this phase here where the membrane actually becomes a little bit more negative than normal. And this phase here is called hyperpolarization. It's more negative than normal. And then lastly, after this hyperpolarization phase, remember that we have sodium-potassium pumps that help to maintain the normal concentration of sodium and potassium around the cell membrane. So the sodium-potassium pumps help to bring this membrane charge back to its resting membrane potential. So at this point in time, I would like to summarize the events that happen during one action potential. Okay, a threshold level has to be reached. The change in the charge has to be significant enough to trigger the action potential. When the action potential occurs, the first phase is depolarization. Sodium ions are moving in during depolarization. Second phase is repolarization. That is when potassium ions are moving out. The third phase is hyperpolarization, when more potassium ions move out, more positive charges move out, and it makes the negative charge inside the cell more negative than normal, about -90 as opposed to -70. And then lastly, the sodium-potassium pump is going to bring that charge back to its normal -70 resting membrane potential. Those are the steps of an action potential. Now, I want to point out a couple of things that are happening with the voltage-gated channels after depolarization and before repolarization. So in this slide, I want to focus on this part of the action potential right here, right at the peak. We've had sodium has moved in, causing depolarization. And then potassium is going to move out and cause repolarization. So what is happening here? Two things are happening. Number one, I want to look at the sodium voltage-gated channels. Down here at the threshold level, the sodium channels had to open to allow the sodium to move in. This is what the sodium channels look like. They have this inactivation gate. So when the charge changed at the threshold level, the sodium channel opened and ions moved through. The sodium moved into the cell. But then once the charge became +30, it triggers an inactivation gate to close. When this inactivation gate is here, no stimulus can make this channel reopen. The inactivation gate is going to continue blocking this channel until the charge of the cell moves back down to a normal resting membrane level. So the sodium channels have an inactivation gate. When that inactivation gate is closed, you cannot have another action potential. The amplitude is always about the same. So this little phase here, phase 3, we call this the absolute refractory period. And that means that no matter what, another action potential cannot occur in that time period. Now we'll look at the potassium channels. When the potassium channel is closed, no potassium can move through. What triggers the potassium channels to open here at this point in time is the charge change. The positive charge of +30 millivolts causes this voltage-gated channel to open. When it opens, the potassium is going to move out, and that causes the repolarization phase. Then we also talked about how we have a hyperpolarization phase. Down here, the charge of the cell is more negative than normal, about -90 millivolts. Now, if there was another triggering event, another stimulus, there would have to be a stronger stimulus to bring the charge of the cell to the threshold level. So at this point in time, once the charge of the cell is negative again, the inactivation gate will come away. It will reopen. And then the sodium channel will just be in a closed position. And then if there is enough of a signal that brings it back to the threshold level, that will trigger the sodium channel to open again. So in this phase, it is possible to have another action potential, but the stimulus has to be stronger. And we call this the relative refractory period. So the absolute refractory period is important because it prevents action potentials from going in both directions along the axon. Action potentials always go from the cell body end to the axon terminal end. And one of the main reasons is because of that absolute refractory period. The absolute refractory period is because of the sodium inactivation gate. Now, what if our axon had myelin? If it was a myelinated axon, then we call it saltatory conduction, and the action potentials happen in a slightly different way. If we look at this diagram of a neuron that has myelin, remember in the central nervous system, the myelin is made from oligodendrocytes. In the peripheral nervous system, these are because of Schwann cells. Now, the only difference with this process is that the voltage-gated channels, the sodium and the potassium channels, are only located in the areas of the axon where there is no myelin. Those areas are called the nodes of Ranvier. So the action potentials, in a sense, jump from one section to another. So when the inside of the cell becomes positively charged because of ions moving in at this end, those positive charges are going to trigger these ion channels to open, and then those positive charges will move in and trigger the next set. So the result of saltatory conduction is that conduction can happen way faster. So when we have those really long motor neurons and sensory neurons, having myelin around the axon means that those action potentials can happen very quickly. If the myelin was not there, the action potentials would likely not make it to the end of the axon. And multiple sclerosis is an example of a disease. It's an autoimmune disease where the immune cells kill the myelin in the central nervous system. And then people would lose motor abilities and sensory abilities. If we look at what we've covered, we have talked about an action potential starting at the axon hillock and moving down the axon because of sodium moving in and potassium moving out, depolarizing and repolarizing the membrane. Now we are at the synapse. Once that charge reaches the axon terminals, now what happens? What causes these neurotransmitters to be released? This diagram is showing a single axon terminal. Okay, we're looking at the presynaptic neuron. It has had an action potential that has moved down the axon, sodium and potassium moving in and out of the cell. When that happens, we are changing the charge of the membrane. We are making the cell inside more positive instead of negative. When that positive charge reaches the axon terminal, it is going to trigger calcium channels. These are also voltage gated. When the charge reaches a calcium channel, it is going to cause those channels to open, which means calcium is going to move in. And when the calcium moves in, it is going to cause these vesicles containing neurotransmitters to move out. And how it moves out, this process, is through exocytosis. When the neurotransmitters move out, they are going to bind to channels on the next membrane. This is now the postsynaptic membrane, and it is going to trigger a reaction in the next cell. So step 4, the neurotransmitters are going to bind to ion channels. What kind of ion channels are these? These are chemically gated ion channels. They are going to open because of a chemical, which is a neurotransmitter. And then step 5, it is going to trigger a depolarization of the postsynaptic neuron. And now the signal can continue. In our diagram, what causes the release of the neurotransmitters? Calcium ions moving in. Calcium channels are voltage-gated channels. They open because of the action potential. The neurotransmitters are going to be released, and when the neurotransmitters are released, they bind onto the postsynaptic neuron. When they bind to the postsynaptic neuron, they're binding to chemically gated channels that will then trigger a response in the postsynaptic neuron. Now, what is that response? This leads us to our final process. What is happening here at this part of the neuron is a graded potential. So we've looked at action potentials, we've looked at the synapse, and now we're going to look at the graded potential, what is happening here when a neurotransmitter binds to that membrane. There are a few differences between action potentials and graded potentials, and it's important to know the differences. Some of the things that we talked about with action potentials, they are all or nothing. They either happen or they don't. they need to have a stimulus that has to reach a threshold level for the action potential to occur. Action potentials happen because of voltage-gated channels. Okay, so keep those things in mind as we talk about a graded potential. One of the things that is very significantly different with graded potentials is that they don't have the same amplitude. Okay, that's why they're called graded. They can have varying amplitude, and that will depend on the amount of stimulus. The other thing that is significant is that stimulus can either be excitatory or inhibitory. There are different kinds of neurotransmitters that will trigger different kinds of ion channels. Okay, so when we go back to our membrane of moving ions across the membrane, if we move positively charged ions in, we are depolarizing the membrane. If we moved a negatively charged ion into the cell, what would happen? What would happen if we moved chloride into the cell? That would make the cell more negative. That would actually hyperpolarize it, and that would prevent an action potential. So let's have a look at how graded potentials work. So here we are looking at our presynaptic neuron. This is the axon terminals, and this is where neurotransmitters are going to be released. Now, multiple axon terminals can impact the postsynaptic neuron. It can be thousands of neurons that impact this neuron. So neurotransmitters are going to be released from this presynaptic neuron, and it's going to act on chemically gated ion channels. The neurotransmitter will bind, and ions are going to move in. So now, these ion channels could be different kinds of ion channels. If it moves sodium in, we know that sodium has a positive charge, and sodium would cause a depolarization of the membrane. If we move positive charges into this postsynaptic neuron, this is going to be the excitatory stimulus that brings our axon hillock to that threshold level, making it from about -70 up to about -55. So when we add positive charges into the cell, that will promote an action potential. So when we change the charge of the cell to positive, we call it an EPSP, or an excitatory postsynaptic potential. However, what if we moved chloride in instead? Chloride has a negative charge. If we move a negative charge into the cell, that will cause hyperpolarization, and that will not promote an action potential at the axon hillock, because negative charges are not going to bring the charge up to -55. It might make it as low as -85. So when we move a negatively charged ion into the cell, we call that an IPSP, or inhibitory postsynaptic potential. So a graded potential can be excitatory or inhibitory, and we can sum those stimuli. If there's enough excitatory stimuli, then you will have an action potential. And if there's more inhibitory stimuli, then you won't have an action potential. And this is important. It's important for our nervous system to balance excitatory and inhibitory. We can't just constantly excite the nervous system, or we would have seizures, or it would not be good. So we have to have a balance between excitatory and inhibitory signals. And it all depends on what the brain and the body are trying to do and how we're responding to things. In the next video, we're going to talk about neurotransmitters, and we will look at some examples of neurotransmitters that are excitatory and some that are inhibitory. So lastly, let's have a look at a summary chart comparing the similarities and differences between action potentials and graded potentials.